Hi, this is Lecture 10, and it's about sex and gender. Now, a lot of what I'm going to be covering in this lecture is material that's also talked about a bit at the end of Chapter 7 in Sapolsky's book, Behave. I also wanted to recommend to you a really excellent podcast that covers topics like today's lecture, but so many other topics that overlap with things that we're going to cover in this course. And it's made by this neuroscientist at Stanford named Andrew Huberman. You can see he's a neuroscientist and a professor in the Department of Neurobiology at the Stanford University School of Medicine. And he has this really great podcast called Huberman Lab that you can watch as a YouTube video. He actually records it as a video, but then you can also download it as an audio podcast. So I check it out. It's got lots of new updated information about a lot of topics. It's mostly been filmed or recorded in 2021, 2022. So it's really up-to-date research about lots of the topics that we're covering in this course. Okay, so what are we going to cover today when we talk about sex and gender? Well, first of all, I want to go ahead and start with an introduction of an interesting empirical finding, and then we'll move on to talk about things like the definitions of sex and gender. We'll talk about sex development and differentiation, which takes up most of this particular lecture. We'll look at some of the activation effects of sex hormones. And finally, we'll look at hormones in utero and sexual orientation. All right, let's start with this empirical finding. Not that long ago, I was at a seminar that was being presented by a professor in economics. And in this particular seminar, the professor mentioned that an article had come out several years ago that had a profound impact on people in economics. And it was this particular article by Needle and Vesterlund called "Do Women Shy Away from Competition? Do Men Compete Too Much?" So it was this interesting gender difference in how much men and women like to compete. And you can read the abstract there. And if you want, you can find the article Quarterly Journal of Economics. So this paper has been cited over 3,697 times, which is quite a lot, given that probably the typical study is lucky to get 100 citations in its lifetime. Well, anyway, what did they do in this study? Well, they had 40 men and 40 women in groups of 4. So they participated in these groups of 4 where there were 2 men and 2 women. They say here in the article that although gender was not discussed at any time, participants could see the other people in their group and determine their gender. Now, the task was they would work on adding sets of 5 two-digit numbers on paper and then put their answer in on the computer. And they did this individually. So each person's just sitting in front of a computer. They might have a problem like 55 plus 32 plus 21 plus 99 plus 11 equals, and then you would go ahead and give that answer after you computed it by hand. And they could see their current number of correct and wrong answers. And they had to do this in 5 minutes, okay? So the first time they would do this, they got rewarded by the number of sets that they had completed. And they would receive 50 cents per correct answer up to $2. So roughly it would take, if you were doing it really well, you could do this 4 times in those 5 minutes and get $2. The second time that they did this, though, they were rewarded by how they did in a tournament of the 4 people. And so they were told that the person who gets the highest number of these solutions in 5 minutes will win $2, but no one else will get anything. And so they did that the second time that they, so the first time they're doing it just individually, they get it based on their sort of piecemeal production. The second time, it's like whoever wins this tournament. Now they're given a choice on the next task. And so on the third task, this third time they do it, they say to the participant, would you like to do it again piecemeal, or would you like to do it as a tournament? Now keep in mind that they've set up the outcomes here so that you could get the same amount of money in a tournament if you actually were the winner as you would do if you got all 4 of these answers correct, right? So if you chose the tournament and you won, you would win the same amount of money as if you had done it individually and gotten all 4 solutions. And what they found was that women chose not to do the tournament on that third task. So despite the fact that on the first 2 tasks, there were no gender differences. You can see that here in the top figure. Piece rate means task 1. You can see there's no difference in between the number of times they make this solution versus for men versus women. And then same thing is true for the tournament. There's no difference in how much they do on that task. There's really no difference here in the outcome between men and women. But when they actually gave them that third task, you can see that twice as many men, 73% of the men, as opposed to only 35% of the women, selected the tournament for the new tasks. That is, these men were much more likely to want to do the third task again as a tournament than women were. Only 35% of them would want to do this task again on a piece rate basis. And how well the participant did in the first task actually had no relationship to whether or not they chose the tournament. And that's being illustrated in figure 2, where you can basically look at if you were one of the worst people on the first task versus the best people on the tasks. You can see that as a woman, it doesn't make any difference whether or not you want to participate in the tournament. Whereas men, you can see that, sure, a few of them who do better actually want to do the tournament, but overall, these men want to do the tournament compared to women. So more than half the men are willing to do this no matter how well they did on the first task. And so what this really meant was that some low ability men probably entered that tournament too much. They were going to end up making not $2, they're going to get nothing. And there were probably some high ability women who could have won the tournament but didn't enter it though. So there's this difference there we're seeing. So why is it that despite their abilities, women just don't want to participate in the tournament? Men want to go ahead and compete. So that was the main finding. This is the thing that really got a lot of attention. And like I said, it's had a big impact in economics about whether or not women are as competitive as men are. So assuming that these findings would replicate, I want you to think a little bit about what's going on here. Is this due to some biological difference between men and women? Is it something about culture? And you might say, well, it could be because women shy away from math problems. But here, the math is fairly easy to do. And you can see that in the piece rate and the tournament and the first two tasks, there is no difference between the men and women on this task. They can equally do it well. So it doesn't seem to have anything to do with their actual math ability, whether that's a cultural or biological difference. This sort of easy task, just women are choosing not to compete. Or is it some sort of interaction, some kind of combination of the two things, the biological and the cultural? So I'll come back to this at the end of the lecture, but I wanted to pose this as a problem. It's a social behavior, competing, and then trying to understand it from the perspectives of sex or gender makes this a really interesting study. All right. Let's move on then to this discussion of sex versus gender. And this is something that is currently very contentious in everyday society, and there's lots of opinions about what sex is, what gender is, and so on. I'm going to try to use as simple as definitions as I possibly can here to be clear about what I'm trying to talk about in this lecture. So for sex, I'm going to say that the term sex is generally refused to a binary of being either female or male, as denoted by attributes that comprise biological sex. So we're going to think of this as more like a biological definition that has this binary output of female or male. Now, even this, as simple as it seems like there's just a binary difference of female or male, you're going to see it's actually more complicated than that because biological sex is more complicated than that. But that's how we're going to treat it. So whenever you hear me talk about sex, I'm referring to attributes that comprise biological sex. Gender, on the other hand, would refer to your sense of who you are as a guy, girl, or something else, as opposed to what your physical characteristics, your genes, and your hormones indicate. So identifying your gender can be more diverse than simply seeing yourself as male or female, and people can express their gender in different ways. So gender has more to do with self-identification. I'm going to consider that more as the way that the participant sees themselves or would identify to others how they see themselves in terms of gender. All right, so that's how we're going to distinguish between sex and gender. Now, if we go back for that experiment that I just mentioned to you, it's interesting here how they're not exactly the most careful about what they mean by sex and gender. They say here, although gender was not discussed at any time, participants could see the other people in their group and determine their gender. So what they seem to be suggesting here is that gender is in the eye of the beholder, that the participants can decide whether or not that other person is a male or a female. They're not asking whether or not that person identifies as a male or female. So notice already we have some sort of controversy about what we mean by gender in this particular study. But with that aside, let's move on then to the heart of this lecture, which is to talk about sex development and differentiation. Now, next week, we're going to be, in the next lectures, I should say, we're going to be talking about genetics and molecular genetics in great detail. And we'll be talking about things like chromosomes. And then you might know already from other classes that the human chromosomes are that we have these 23 pairs. And it's the 23rd pair where we talk about the differences between female and male genetic differences. And so we'll say, as you can see here down in the lower right corner, we have an X and a Y chromosome. Females typically have two X chromosomes, whereas males have an X from their mother and a Y from their father. So the female genotype is XX and the male genotype is XY. Notice that the X is larger and it contains several thousand genes, whereas the Y chromosome only contains about 100 to 200 genes, a lot smaller. And only about between 45 and 73 of these are actually protein coding, which you'll see in the next lecture is an important distinction. How many of those genes are actually protein coding will make a difference for behavior and structure and so on. All right, so that's sort of the typical way that you think about these chromosomes. That in this picture, obviously this is a male because the male would have an X and a Y chromosome in terms of chromosomal sex. Now that means then we could actually talk about sex biologically in three different ways, and I'll kind of bounce back and forth between these in this lecture. So obviously we have chromosomal sex, where XY would indicate that the person is male based on their chromosomal sex. We have XX, which would be a female, but there are also some people out there who have XXY or XYY. So it's important to keep in mind that it's not just simply this binary thing of XY versus XX. There are a few people out there with XXY or XYY, and there's a lot of implications for having chromosome patterns like that. Won't be getting into that in this lecture, but I refer you to other texts and other lectures about those kind of people. Now, another way we could define sex biologically is based on the gonads, gonadal sex. And so if a person has testes, we would say then they're male based on their gonads. And if they have ovaries, we could say biologically they're female. So that's another way to determine sex biologically is by looking and seeing are there testes there for males, ovaries for females. And finally, there's another way that we could define sex biologically, and that's in terms of hormonal sex. That is, if it's primarily testosterone, or I should say androgens, we could say the person is male. But if their hormones are primarily estrogen versus androgens, we would say they're female. Now, these three distinctions, these two different ways of defining sex biologically, aren't always going to be one and the same. That is, for most people, chromosomal sex, gonadal sex, and hormonal sex are consistent. That is, if you have an XY chromosome, you're probably going to have testes, and you'll primarily have testosterone as your primary hormonal sex versus the estrogens. But sometimes these don't line up. And as I talk later in this lecture, you're going to see places where the chromosomal sex doesn't necessarily match up to the gonadal sex, and the gonadal sex doesn't necessarily match up with the hormonal sex, and vice versa. All right, so now that you kind of think of these different ways that we could define sex biologically, let's now look at the development of the reproductive organs. So when the sperm does get to the egg, and we now have a fertilized egg and an embryo starts to grow, it turns out that in the first 6 weeks of an embryo's birth for humans, it's a sexually undifferentiated embryo. So for the first 6 weeks, you can't tell if it's going to be a male or a female biologically. In fact, if you look there up at the top, you can see that's what it looks like at first, that we have these ducts that are formed, and they're uncommitted gonads. There are two ducts, the Mullerian ducts and the Wolffian ducts. And right now, they're both existing in that embryo for at least for those first 6 weeks. And then as the fetus develops, what's going to happen is, if the fetus has a Y chromosome, and on that Y chromosome they have the SRY gene, which we'll talk about at a future lecture, it means then that you're going to get testosterone. And the testosterone then causes Wolffian duct to develop into male internal organs. So you can see that on the right, If the fetus has a Y chromosome and the SRY gene is on that Y chromosome, which we'll talk about at the next lecture, this would mean that you'll get testosterone, right? So you're going to get these androgen hormones. And they're going to cause then that Wolffian duct to develop into male internal organs. At the same time that it's developing, it's going to cause the Mullerian duct to become inhibited and to degrade. And so you can see that on the left there in blue, that the Mullerian duct is degrading and the Wolffian duct is becoming more prominent, and eventually it's going to disappear altogether. So, I'm sorry, the Mullerian duct is going to disappear altogether and the Wolffian duct is going to become the testes, right? And then the same thing happens over on the female side in the opposite way, where you're going to see that, well, if without any testosterone there, you're going to get the Wolffian duct degrading, and then the Mullerian duct is going to become the tubes in the uterus for the uterine tubes in the uterus. And so you can see there on the red side, that's what happens by default. So what this really means is sort of by default, our bodies would normally want to become feminized. They're going to become the path that you see on the right. But if the SRY gene is there on the Y chromosome, it's going to cause testosterone to be formed, and then that hormone circulating is going to cause the Mullerian duct to degrade and the Wolffian duct to actually become prominent. And so it undoes that sort of default of feminization of the sex organs. And also, I just mentioned that if you have XX chromosomes, that is, you're an XX female, this would also mean you're going to have increased feminization of the brain and body and more demasculization. So what happens is it's not just that it all becomes feminized, it's becoming more feminized in the XX chromosome configuration. Now, at this point, I just want to mention some terminology that kind of gets slipped in here and now and then. In fact, I think I might have even brought some of this up in a previous lecture about adolescence. That just want to point out that when we talk about primary sexual characteristics, we're talking about the ones that appear at birth. Okay, so the ones that you can see easily when a baby is born are primary sexual characteristics, like the penis on a boy and having ovaries and vagina in a girl. This would be primary sexual characteristics, the ones that appear at birth. What we call secondary sexual characteristics are the ones that appear at puberty. Okay, so things that change at puberty, new things that occur on the body at puberty are going to be known as the secondary sexual characteristics. Now, let's go ahead and put a spotlight on testosterone. Okay, now testosterone is going to have effects on both male and females. Let's look at it though in terms of XY males. So males who have that XY chromosomal pattern. It turns out that in XY males, it's not the testosterone that causes the penis to develop. Right? So you might think that, as I just got done telling you, that the testosterone had a lot to do with the organs becoming more male rather than female, but it turns out that it's actually another androgen, dihydrotestosterone, or DHT, that is this dominant hormone in males that causes the penis to develop. It turns out that we have DHT in our bodies all of our lives, and it's responsible for aggression, muscular strength, beard growth, male pattern baldness. Okay, so dihydrotestosterone is there, the DHT, this other androgen. In the fetus, testosterone is converted by an enzyme called 5-alpha reductase to DHT. So the testes create testosterone, and then the testosterone is then converted from this enzyme to DHT. And it does this in a little structure called the tubercle, which will then develop into the penis. So it's the DHT that actually causes the penis to develop. Then in puberty, hormones will be released by the pituitary gland to cause testosterone to be produced. And then at that point in puberty, the testosterone is going to cause further development of the penis and all those secondary characteristics that we talked about, for example, in adolescence. So that's sort of an interesting thing. You would think it might just all be testosterone, but it turns out the DHT has this important role because of this 5-alpha reductase converting the testosterone to DHT, that we actually end up having this male genitalia. Okay, now to show you how complicated this can actually get, I'm going to go ahead and tell you about a case of what are called the guavidoses. Okay, and in some people, there's a genetic mutation that causes the 5-alpha reductase not to be produced. Okay, and this was first identified in the Dominican Republic, so that's why they refer this in Spanish to guavidoses. What happens is the baby is born and appears female, so they'll have like a ceremony to celebrate. They have a girl, and there's no external penis, so they don't think that this is a boy at all, and they start raising that baby as a girl. And then around 11 or 12, though, a penis suddenly appears. And what goivodosis means is that means basically literally penis at 12. Okay, so they found these cases of goivodosis in the Dominican Republic because, like I said, it's a genetic mutation. It seems to be passing through in some families there in the Dominican Republic, but it's been since found in other parts of the world as well. All right. So what this means for these particular individuals is they were actually born with testes, right? Their testosterone was released, their Müllerian and Wolffian ducts followed the patterns of a male development, but it turned out that the testes itself didn't actually create a penis that was external. It was stayed internal. It didn't descend, okay? So it's up inside that they have this penis that doesn't become developed because there is no 5-alpha reductase being created because that enzyme isn't produced due to this genetic mutation. Therefore, no DHT is created, and therefore the penis didn't develop. But as I said, at the puberty, what happens though is that testosterone is then released, causing the penis to develop further. So this penis that wasn't actually developed during fetal development suddenly now does develop, and boom, you now have a penis on this child that had been previously thought of as a girl. These two children that you see here both have goivodosis, they're cousins, and the girl on the right has already been documented to have this condition that she'll be, when she reaches puberty, she'll actually have a penis. So it has all sorts of interesting social and family ramifications because of the way this happens. But again, I wanted to point out, this just shows you then that having a mutation of like this 5-alpha reductase not being created then is going to cause that DHT not to be converted from the testosterone. And therefore, early on, when the penis was supposed to be developed by DHT, that didn't happen. But during that time of puberty, there's testosterone coming in and saying, all right, time to make that penis develop further. And now the child actually has a penis when before didn't seem to have one at all. Okay, well, that's goivodosis. Let's go ahead and continue talking about testosterone and talk about how the brain is not actually masculinized by testosterone in XY males. This is an interesting finding as well. Turns out that male and female brains have receptors for both testosterone and estrogens, okay? So both male and female brains have receptors that mean that in different parts of the brain that testosterone and estrogens are going to bind. Testosterone can be converted into estrogen by the enzyme aromatase. All right, so you're pumping out testosterone from your testes, and what will happen is this enzyme aromatase will then convert it to estrogen. So it's another way of taking testosterone and converting it into something else, this time estrogen. And what happens then in puberty is that the testosterone goes up. As I said already, testosterone is going to increase in production during puberty in these XY males. And it turns out that some of these males will actually start growing breast buds or small breasts. And this also sometimes happens to bodybuilders who are taking super amounts of testosterone. And the reason is because of all this excessive testosterone that's being created. A lot of it gets converted by the aromatase, this enzyme, into estrogen. And now all of a sudden you have all this estrogen out there, and the estrogen now binds to the receptors in the brain of these XY males and causes them to have secondary characteristics that look like what you'd expect in an XX female. That tells us then that really it is really quite important that we have both testosterone and estrogen in our development of the fetus. And it turns out specifically that the brain's wiring for a more masculine brain is done mostly by this converted estrogen. So why aromatase converts the testosterone to estrogen is because that estrogen is eventually going to be what sort of wires up the brain to make it a more masculine brain versus a more feminized brain. And when I talk about a masculine brain versus a feminized brain, I'm talking about having certain characteristics, again, that match onto the biology of an XY male versus an XX female. Whether or not this all lines up perfectly, that we get, again, that lineup between chromosomal sex and hormonal sex and so on, we don't know. It's not necessarily going to all line up perfectly, and that could explain why there's a lot of variation in the way that people think of themselves. But in this case, it's really the estrogen that actually is doing most of that wiring of the masculine brain. And also, of course, it's helping with the wiring of the feminine brain. Now, continuing to talk about testosterone, let's talk about something called androgen insensitivity syndrome. Now, this is another genetic mutation. it's due to genetic mutation, where people who have XY chromosomes look like and appear like, and feel like they themselves are females. And the reason for this is that because of this genetic mutation, the receptors in the body for testosterone are missing. So now we have cells in the body that normally would have had testosterone receptors there that would cause changes in the organization of the body. But because the receptors are missing due to this genetic mutation, the testes don't descend. So again, much like those guaivadoses, you're going to have people who develop the testes, but it keeps inside, it's internal, doesn't descend because the receptors are missing. Okay. And so because of that sort of thing that would happen then in puberty, testosterone in puberty doesn't actually change the secondary characteristics because the receptors aren't there. So the person ends up looking more female. So in this case, the person in all other ways, because their testosterone receptors aren't there, they have female characteristics, they have breasts, their hips change, they don't have facial hair. All those things that testosterone would normally be doing to a boy doesn't do this to an XY chromosome person who has androgen insensitivity syndrome. So what in this case means then in the end is that their chromosome and their gonads are actually male. There's no way that they can actually get pregnant because they don't have the right gonads for that. But otherwise, hormonally, they're being primarily driven by estrogen since the testosterone receptors aren't there. So hormonally, they actually really have a female profile. And so, like I said, for all their lives, they can basically be female, look female, and feel female, even though they have actually these undescended gonads and they have an XY chromosome. So these are all sort of interesting things to show you that it's how the hormones and genetics play a role here through mutation, through the hormones. We can get really quite interesting combinations of things depending on how we want to define biological sex. Let me just mention some other external factors that can affect more estrogenic versus androgenic development. So what I'm saying here is that, if we talked about just hormonally, you having more of a female profile, it would mean that you're primarily being driven by estrogenic development versus a more androgenic development where it's more things like testosterone that are affecting the development of that person and their behavior. Here are some interesting other factors that can influence that. One of them is something that just happened in the last 10 years or so, and it's this problem or these findings associated with evening primrose oil. Now, evening primrose oil is used for sort of like skin products for skincare and other kinds of products with cosmetics and so on. And it turns out that when you rub it onto the skin, it actually has estrogenic effects. So in the primrose oil itself, just the way it naturally occurs, it takes on the structure that's very close to estrogen. And it gets rubbed into the skin and it sort of gets absorbed then into the skin, into the bloodstream as if estrogen's being released. And what they found was that boys who maybe were hugging or touching a female like their mother or their sister who had just rubbed on primrose oil were developing breasts. And it could be what the stated effect was. It turns out that the estrogen effects of that primrose oil were being rubbed off through the skin onto that boy and causing that boy then to have these more estrogenic development because of the evening primrose oil. A similar thing happens with testosterone creams. There are a lot of people who actually use testosterone creams to have more androgenic development. They're trying to lean their body more towards this testosterone developed body. And so they apply testosterone creams to their skin. And again, the same thing could happen when you rub it off onto somebody like a girl. And it turns out that maybe that testosterone is going to promote androgenic activity in the girl and change some of her features that way. There's also something that is not a product that we would normally pay for called atrazine. This is actually found in herbicides, and it's been found in lots of laboratory studies and in frogs that it diminishes testes development. Okay, so this additive that's in herbicides, if it gets consumed, it diminishes testes development. And what that's doing is it's really causing sperm counts to decline, probably due to this atrazine in herbicides. And you could point to the declining sperm counts in recent decades in the US and Western Europe, where a lot of this research has been done, as having to do with the fact that atrazine is appearing more and more in herbicides. It affects the developments of the gonads and the brain, and it probably is also contributing to the early puberty onset in girls that's been found in these cultures. So there's an external factor again, atrazine, that's found in these herbicides. And as a result of that, it's actually affecting gonad and brain development in boys and girls. And another external factor that has a role here playing in estrogenic versus androgenic development is cannabis. There are more studies now that are showing this. It's not really clear yet if it's the THC or the plant leaves themselves that cause this to happen, but basically what cannabis does is it increases aromatase. And you remember that aromatase then can be used to have more estrogenic effects, right? So aromatase takes your testosterone and converts it to estrogen. And so with this increase of aromatase in somebody who's doing a lot of cannabis, it promotes more estrogen to be released. And this even happens prenatally. So if you have a mother, for example, who smokes a lot of pot, she can actually cause more aromatase to be formed, thus having more estrogen influence on the developing baby, and therefore it will affect the baby's primary and secondary characteristics. And keep in mind that this baby could have an XY chromosome or it could be XX. And so the estrogenic effects could be enhancing or inhibiting some of the effects of the chromosomes themselves. And one more external factor to bring up is alcohol. It turns out that alcohol, especially grain alcohols like beer, also increase estrogenic activity, especially during puberty. So alcohol, when it's imbibed and gets into the bloodstream, it causes more estrogen activity. And therefore, if you're doing a lot of alcohol, apparently, it's going to cause more estrogenic development during the puberty years. So these are all factors that, like I said, you wouldn't say that this is all just being determined by your chromosomes, about whether or not you have an XY or an XX. It's really kind of showing you how much the environment also has a huge influence then on our development, our sexual development, whether or not we're going to become more male-like or more female-like in terms of these sort of binary divisions here in the biology of sex. All right, let's move on to the fourth part of this lecture, which is to talk about activation effects of sex hormones. Now, you might remember that hormones have these two big effects. They have organizing effects where they could affect changes in the structure of the body, like having the penis develop or to have breasts develop or whatever it is. But there also are these activation effects that can have sort of immediate effects on behavior. We've already talked a bit about some of these things. We talked about, for instance, how testosterone might have something to do with competition and so on. Well, let's look a little bit more at how these activating effects might affect sexual behavior. So for both males and females, it turns out that estrogen is really important for libido. Both males and females have to have the right amount of estrogen in order to have enough sexual desire. If estrogen levels are too low in males, they won't have as much of a libido, much of a sexual desire. So it's interesting. You might just think that only testosterone would have something to do with libido in males. But it turns out that estrogen is also quite important for libido. Now, keeping with males, turns out though that testosterone is going to be important for sexual excitement. So sexual excitement is highest when testosterone is the highest. And this is sort of the peak time is between 15 and 25 years is when testosterone levels are the highest in males. And by the way, again, for males, I mean in XY chromosome males, but I think a lot of these studies are actually done on people who identified as male. And so we don't know exactly how much of this is chromosomal, but keep that in mind as a caveat. Sexual pleasure during orgasm is interestingly related to the secretion of oxytocin in males. So during that sexual pleasure, you get this oxytocin, which you remember, oxytocin may be sort of like a social hormone that helps us bind in relationships. So it kind of makes sense that it would be secreted during orgasms. And then finally, decreased testosterone levels generally leads to decreased sexual activity. So if you have stress or age or something else that's going to cause the testosterone levels to drop, you're generally going to find decreased sexual activity in males. Now, what about in females? Well, the thing that a lot of researchers focus on in females is the menstrual cycle, because there's all these changes that are going to happen monthly in terms of hormone levels. So we can take days 1 through 5 of the menstrual cycle and just talk about menstruation as the beginning of it. And this is when the inner lining of the uterus or the endometrium partially disintegrates, okay, and is going to be passed out of the body. Then during days 5 through 15, we have what are called the follicular phase. And this is when the anterior pituitary is releasing follicle stimulating hormone, or FSH. And then it causes follicles then to release estradiol, an estrogen, which increases the production of FSH. So it kind of feeds back to the anterior pituitary to keep creating more and more FSH, and also creates a hormone called luteinizing hormone, or LH. Then, during days 16 to 28, you get what's called the luteal phase, and the FSH and the LH cause the follicle to release an ovum, or an egg. And then the remnant of that follicle is going to release progesterone. Okay, so the egg is going to be released by the follicle itself is going to release progesterone. If the ovum is fertilized right there at the beginning of the luteal phase, then estradiol and progesterone continue to increase through pregnancy. If the ovum is not fertilized, then all the hormones just sort of decrease in their production and go back down to baseline, back to where we are at days 1 through 5 through menstruation. So we have these 3 different hormones that are changing through, or actually 4 hormones that are changing during the course of the menstrual cycle during 28 days here. And you can see if I kind of map down here where you can see at the top there, the anterior pituitary hormones of FSH and LH, they sort of peak there near the end of the follicular phase. Then we have estradiol that's being created because the FSH has started to go up, and now estradiol is being created by the ovaries, and the estradiol starts to peak and go up. And then finally, as I told you, when the follicle releases its egg, then the ovum, the progesterone is going to increase after that. So if you kind of look down at the bottom there, you can see these different phases I just talked about. There's menstruation, the first 5 days, then we have that follicular phase, and then we have the luteal phase. The thing that's probably most interesting for research in social psychology and social neuroscience is that para-ovulatory period. So it's around the time there near the end of the follicular phase, and we're getting sort of this rush of LH and FSH, and also resulting, they're getting estradiol going up, that we are the most fertile. So females are the most fertile during this peri-ovulatory period. Does that have anything to do with anything? Does that seem to change behavior because of that? Well, we'll come back to that in a moment. Let me just say, though, there are some other things that we could say about females. That birth control pills, by the way, prevent the surge of the FSH and the LH by the anterior pituitary. And so estrogen progesterone pills make it harder then for the sperm to reach the egg, or the egg to be implanted, because the production of FSH and LH isn't actually happening. Then I just mentioned there was that peri-ovulatory period. That's this midpoint of the menstrual cycle when sexual interest and activities increase, perhaps due to higher levels of estrogen. Okay. And then finally, one other thing we just want to mention is premenstrual syndrome, or PMS. Somewhere between 2 and 10% of women experience really serious problems or symptoms that interfere with their work and school, etc., because of these changes in these hormones. And so that's also something that could be affecting behavior. Now, let's go back to this question about whether or not the peri-ovulatory period means that there's going to be any changes in the behavior. For a second, let's just drop to non-humans and look at them. Turns out that a lot of non-human species really have these seasonal variations in which they'll mate, in which they're going to breed, right? And it turns out that a lot of that's being regulated by this biological clock, the suprachiasmatic nucleus that we talked about a few lectures ago. And melatonin production is affected by the amount of darkness, right? And then what happens is, because the eyes are picking up more light as we get out of winter and into the springtime, the days are getting longer, there's more light. The suprachiasmatic nucleus is actually sensitive to this and senses that there's a change in the calendar. And as a result of that, hormones then become released. The melatonin is then inhibited, and then you get increased fertility. So during that time, it particularly becomes a fertile period for a lot of different animals. And this typically is going to happen around spring. So that's why you get so much mating behavior happening in the spring. And it makes sense from an evolutionary perspective. Spring would mean then you've got this several months ahead, where there'll be a lot of growth of plants and other animals that are out and available. There's plenty of food. And you wouldn't want to necessarily get pregnant just before the winter comes on, right? So that's why you would get this seasonal variation that a lot of other non-human species have. But what about humans? Well, humans don't actually only mate in the spring. We don't have these seasonal variations, but we may have variations that are more on the monthly clock, the one that follows more the menstrual cycle. And so some interesting research has been done in this area. Here's just an example of a study that was published in 2007 by Marty Hazelton and her colleagues. You can see that the title of this paper is Ovulatory Shifts in Human Female Ornamentation. And now what human female ornamentation means is she's talking about specifically women's clothing. So the phenomenon she's talking about is, can we try to understand when a woman dresses to look more attractive versus less attractive? Is there any sort of relationship between the clothes that she picks out to wear every day and where the woman is in her menstrual cycle? Perhaps it's something like, the way those non-human animals suddenly are now ready to be bred at springtime. Maybe there's something that happens like this in the monthly menstrual cycle, the ovulatory cycle. So they argued in this paper as their main hypothesis that women dress most attractively during the fertile phase of her ovulatory cycle to attract potential mates. So they're saying that we basically have this basic mechanism that women want to become impregnated during their most fertile phase. And so that's when they're going to try to dress most attractively. They're going to use clothing that does this. So when the method that they used in this paper was they took 30 partnered women at UCLA, and they had them pose for a photograph during a high fertility day of her cycle in the follicular phase just prior to ovulation. So during that time period there where it's actually the most likely that you could get pregnant, right? And then they also picked a low fertility day, which was 4 to 10 days before the onset of the next menstruation. So the other day wasn't during the time that they were menstruating, it was just another day where they couldn't actually possibly really get pregnant because the fertility levels would be so low. So in this luteal phase. So you have two photos, once during the follicular phase, once during the luteal phase. And of course, they don't tell these women how to dress or anything like that. They just say, take a picture of yourself and bring it to us in the lab on those 2 different days. And then they had participants look at those pictures who were blind to the hypotheses of the research. They had judges, 17 men and 25 women, who then looked at the 2 photos and said, and were asked to indicate, was the woman trying to look more attractive in one picture or the other? So they would pick which picture looked more like they were trying to look attractive, right? And what they found is nicely shown here in this figure, that the closer you got to ovulation, so the day of ovulation is where it says 0 down there in the lower right corner. In those days leading up to ovulation is when you're going to have the most fertile period for a woman, when she can most likely get pregnant. As she gets closer and closer to that day of ovulation, you can see that the picture that they were more likely to pick was the one from that period of time. So if they picked, they're comparing 2 photographs, one during the luteal phase and one here during this days during ovulation, you can see that they're more likely to have picked a photograph increasingly as that comes from that period when they're most fertile. And so the relationship here was that they did get some support for their hypotheses here that women were more likely to look attractive on their high fertile days versus their low fertile days. Now you might say, well, what exactly are they doing in these photos? Well, here's a table from that article where they asked the judges to indicate things like, were they looking more fashionable? Did they have nicer clothes? Were they showing more skin in their upper body, showing more skin in their lower body, and so on? And you can see that on the high fertile days, they actually are more likely to be wearing fashionable clothes, nicer clothes, showing more skin, wearing sexier clothes. Well, wearing sexier clothes, you can see, isn't as often as the low fertility days, but it still was quite common among people in the high fertile condition. But anyway, so you can see the kind of differences then between high fertile and low fertile photographs. So suggesting that even in terms of the ovulatory cycle, that human females do change their appearance perhaps to attract mates because just like the way these other animals might do on a seasonal basis. Now, if that's not very convincing, let's look at another kind of effective ovulatory shift on women. And this one has to do with their preference for masculine faces. Okay, so this is a paper that was originally published in Nature in 1999. And what they did was they showed their participants pictures like this. They are actually morphs of several photographs of male and female faces. So they've been morphed together. And what you can see there, that the pictures on the left column, all 3 of those, are pictures where the photo has been manipulated to be more feminized. So there are more feminine pictures going into that face than the ones on the right, which tend to have more males into the face. So the pictures on the right are considered more masculinized. The faces on the left are considered more feminized. And what would happen is the participant would see a pair like on each row there, and then determine which one they thought was most physically attractive. So of those two pictures, which one do they find to be most physically attractive? Now, it turns out that in most cases, and other research had already shown this, that you're more likely as a woman to choose a male who looks slightly more feminized. And so you can see that here in B. So in this study, which was in B, was actually conducted with Japanese participants. These Japanese women were more likely to choose a face that was more feminized than they were to pick a face that was less feminized. And so you can see then that those pink bars mean that they're picking faces that are more feminized than the faces that are not. Okay, so that means that they're more likely to pick slightly feminized faces. But over on the right, what you can see there is we got British women choosing between these two faces. And there were two different IVs here, two different independent variables they looked at. One is whether or not they're on the pill. So you have on the pill, low conception, or high conception. So if they're not on the pill, that would mean that they have a high chance of getting pregnant. Low conception would be that they're not taking any birth control right now. And they were supposed to imagine looking at this pair of photographs and saying, okay, let's just say you're interested in having a short-term hookup with this person, something sexual, okay? Which face would you pick? And you can see there's a slight preference again for feminized faces on a short-term context, if there is no chance of really them being conceived. But if there is a chance that they could be pregnant, you can see that high conception risk is the blue bar. You can see then that those people actually are less likely to pick a feminized face. So they're more likely to pick a masculinized face. And then the other one you can see there is the long-term context. This would be, you're thinking about this person as a potential long-term partner, somebody you could have kids with in the long term. And you can see then again that the conception risk, the low and high conception risk, this time there's no difference between the two of them. There's a slight increase here, a slight preference for more feminized faces. So the idea is that perhaps what this research is suggesting, that a woman who's in a position where her body is basically most fertile is more likely to pick a more masculinized face, even though the rest of the time she'd be more likely to pick a more feminized face. And you might say, well, why would they want to be interested in a feminized face in the first place? Well, because a more feminized face might have features that are associated with taking care of children, taking care of you. They're not going to be so aggressive or tough, but it turns out for sexual reasons in a short-term concept, this is suggesting that you would actually, this woman would actually prefer to have a face that's less feminized. Now, that was the initial study in 1999, and I've given you the reference here, so you can go back and look at it if you want, if you're really kind of unclear what I'm talking about. But the more important point I want to make here is that there's been many studies done since then. And this was a nice meta-analysis that was published in 2014 in a leading journal called Psychological Bulletin. And they looked at all sorts of studies that have looked at these similar kinds of effects for females and their ovulatory cycle. And what you can see here is that compared to low fertility days, women in high fertility days across these studies would prefer men who had facial masculinity, that they tend to prefer men with torso hair, that they have facial symmetry, that they're tall, that they're aggressive, that they're arrogant, that they have a more male typical walk, and have more vocal masculinity. They have like a deeper masculine voice. And what this table is showing you is all these different studies. And again, you can go look up the article if you'd like to see all of the different studies. But the important part is that the thing that's going down the middle there, that's the band. That's during the peak fertility part. And those are the places where you can see in the dark box there, where you're finding the preferences for masculinity, for torso hair, for facial symmetry, and so on. On the rest of the cycle, there's not such a preference for it. So it only seems to be during this peak fertility period that women tend to prefer all of these other characteristics. But when they're not in their fertile period, they're not as interested in all those characteristics. And it's actually been defined there by those gray boxes that you see. So interesting relationships between the ovulatory cycle and social behaviors and mating choices. Now, we will talk more about this in future lectures. We have a whole lecture where we actually talk about attraction and sexual mating, and we'll be talking about evolution and so on, where you can kind of give you more context for these kinds of findings. But I just wanted to give you a flavor right now about how these activating hormones, these things that are changing on a monthly basis for females, could actually have some ramifications for their social behaviors. All right. The last thing I want to talk about is hormones in utero and sexual orientation. Now, this is talked about again in the Sapolsky book a little bit, so I just went ahead and just review with you a little bit about this. It's sort of interesting research, and this is really talking about how the hormones that are presented to the fetus that's developing before they're born, okay, may have long-term ramifications for sexual orientation. Now, here's sort of how this story played out. So back in the 1990s, a researcher named Dennis McFadden at the University of Texas was studying the auditory system. Okay, so he wasn't actually interested in sexual behavior, but in the process of studying the auditory system, he had uncovered this thing called otoacoustic emissions, which are sort of a strange thing, but basically your ear can actually make soft, tiny little sounds while you're listening to stuff, okay? And they discovered while they were studying the auditory system that young males had more of these emissions, more of these sounds, than young females. There's a huge difference between males and females in terms of their otoacoustic emissions. Now, they also found that women who identified as lesbian had more otoacoustic emissions than females who didn't identify being lesbian. So this is sort of an interesting thing. It's saying something that has nothing really to do with sexual behavior, the sounds that you make out of your ear while you're listening to stuff, actually seem to have something to do with sexual orientation and even gender there when you're talking about young males versus young females. Now, interestingly, before this period, there had been lots of other research looking at hormones and seeing if there were differences between homosexual and heterosexual participants. In particular, there had been a lot of research looking at testosterone levels. When they could start measuring testosterone, one of the first things they started looking at is there a difference in testosterone levels between heterosexual and homosexual males. And in most of those studies, they didn't actually find any differences in testosterone levels. And in fact, what they did find sometimes was that gay men often had higher levels of testosterone, which didn't seem to fit with any theories that people had about what would cause homosexuality. After Dennis McFadden had found this study with otoacoustic emissions, then we have in 2000 Mark Breedlove, who discovers that the finger length ratios of homosexual and heterosexual participants differed. And they published this paper in Nature, and you can see the title was Finger Length Ratios and Sexual Orientation. What they did in this study was they went around and measured people's digit ratios. So they measured their 2D and their 4D, or their D2 and D4. So the D2 is your this finger, your index finger. 4D is your ring finger. And what they do is they measure your 2D and your 4D, the lengths of those, and then express it as a ratio. And so if your 2D was exactly the same length as your 4D, then you would find that the ratio would be 1.0. If the 2D is just a little bit shorter than the 4D, then it's going to be a number that's less than 1. So like in this example, it's 0.945. Now, on average, it turned out that the D2-D4 ratio was greater in people who identified themselves as female. Okay, so that would mean that closer to 1 would be a D2-D4 ratio in females, people who identify as female, than in people who self-reported as being male. And this was particularly so on the right hand. It can actually happen in the left hand, but it was more reliable on the right hand. That's nothing to do with it, by the way, with handedness. Okay. So again, this would mean that females are closer to having similar lengths than people who identify as being male, where that ratio would be lower. Now, this is not only the case that this happens in humans, it's also true in mice. They found that in mice and in humans, the more exposure to androgens in the uterus, in utero, the smaller the D2-D4 ratio. That is, the D2 is slightly smaller at birth if you've had more androgen exposure during development, during fetal development. So in mice and humans, the more exposure they have to androgens, the stuff that kind of leads more to testosterone, the more supposedly masculinizing of the brain and the body, the smaller that D2-D4 ratio at birth, right? So this isn't anything that's going to be a function of experience or the environment. It's just already at birth, it's happened. And you've already got this exposure to the androgens in the uterus. Now, why all of that is interesting is then there was this subsequent finding, which that with self-reported lesbians have also a smaller D2-D4 ratio than straight women. So again, in people who identify as being lesbian, their 2D is slightly shorter than their 4D. So that means that they're going to have a smaller ratio than women who identify as being straight. So that's interesting. So something at birth is related to whether or not these people later in adulthood report as being lesbian or straight. Now, interestingly, sexual orientation differences was less clear in males. So you don't find this as reliable for looking at gay men versus straight men, but it does seem to show up with self-reported lesbians versus straight women. And there's no real data. No one's done really much on bisexuality, so we don't really have anything about bisexuals in this data. So again, this is really interesting. It's trying to show us that some organizing effects of the hormones possibly is related to what happens later in life. Who would have thought the otoacoustics would actually have any relationship to anything sexual? But this is just some food for thought, some interesting things. But keep in mind that lots of people since then have tried to do a lot of this D2, D4 ratio stuff. And I would have to say that it isn't really predictive of anything. That is, you can go ahead and measure someone's 2D, 4D ratio. You could look at it on your own hand. You can look at it in your friends. But you'll probably find that it doesn't predict anything. It's just something that only shows up when we have large numbers of participants. So it's a very small effect, and it only seems to show up when we're looking at a big population or large samples of people who identify one way or the other. All right. So that's the final thing I wanted to mention that comes out of the Sapolsky book. Let's go back to that empirical finding now, just to have you, to end this lecture, to think about that study where women shying away from competition, do men compete too much? And I mentioned again that this is a highly cited study, cited over 3,600 times, so it's really had a big impact. So again, I'll raise these questions. Assuming that these findings would replicate, what's going on? Is it biological? Now, you could think that you could make maybe a biological argument here, that there's something to maybe have something to do with the way androgens and estrogens affect the brain of males and females. That's something to do with maybe during fetal development, there was something maybe about the way these things affected risk-taking and later in life. And then again, you could argue that perhaps it's all cultural, that perhaps culture raises boys to be more competitive than girls. And into looking at that question about culture, we're going to be talking a lot more about culture in lecture 13. And then it also could just be a bit of both. It could be some sort of interaction or something that happens here when we look at sex or gender in this case, and then see how it possibly interacts with different kinds of cultures that might be more competitive versus more cooperative. So all very interesting questions. I don't know what to make of it. I don't know if I would want to say that anybody thoroughly understands these particular kinds of results, but I'll leave you there on that. So in this lecture, what we did is we talked about that empirical finding about women being less competitive than males. We've talked about the differences between sex versus gender, and we talked about a lot about sex development and differentiation, the activation effects of sex hormones, and finally, hormones in utero and sexual orientation. I specifically talked about those otoacoustic and the 2D, 4D ratio stuff. So thank you very much, and I'll see you at the next lecture.